Nearly a century and a half ago, the big news about encoding and playing back sequential information, the forerunner of movies, was all about those horses. Over the years, the technology advanced to motion picture film, and then to tinier and tinier videotapes and flash media. And we thought that was gee whiz stuff. Well, think again. Today's latest news about encoding and playing back sequential information is again about those horses, except these movies were encoded in and played back from DNA in living bacterial cells.
To be totally honest, the point is not to store videos in bacteria. We used the video because it's a good example of a complex piece of information that has both many parts to it, that is many pixel values, and a time component. So it's organized over time. And so it was a good way for us to test whether the CRISPR adaptation system that we're using could actually acquire enough information that we could go in and sequence the bacteria after we had encoded it and reconstruct the movie. Right? So it's kind of a way for us to push the technology forward and understand how we will build and analyze data as we are scaling up to hopefully eventually do biological recordings. My background is actually in neuroscience, and so I was trying to understand how different cells in the developing brain would become very distinct types of neurons. So they all start as a cell with the same genetic material, but over time they acquire these very distinct, we call cell fates, so the different types of neurons. And it's very difficult from a technological standpoint to understand how that happens in the developing brain, because it's happening for many, many, many cells at once and all kind of locked away within the brain. And so to get access to the biological events that are happening is quite difficult. And so what I imagined was, if we had a way of recording biological events and storing them in the genome, then I wouldn't have to, as an experimenter, go in and either be watching the cells or be kind of disrupting the brain in order to get information out, which then disrupts the entire developmental process. The overall idea is to use these kind of molecular recorders to gain access to biological phenomena that we just can't see in real time. And I was making reference to the development of the brain, because that's one place where it's something that happens both over time and in a biological system that's very difficult to get access to actually watch the cell biology occur. So if we were able to implement a molecular recorder to catalog biological events over time, that's the type of system that really would, I think, a molecular recorder would fundamentally change the type of data that we would pull out of the system. Right now, we're really piloting the idea of encoding information into the genome as a way of storing information in live cells. And so, it's in bacteria. We're currently supplying the information, and over time, we really would want to kind of unlock this technology, be able to put it into any cell type, and to hook it up to biology. It is now functioning in bacteria, and so there are other things you could think about, like having living bacteria that are actually sensing things in their environment and recording them into their own genome. And so they could be kind of these biological recording devices, sense and recording devices. And so they could be out in the environment, or just recording things that are happening within the cell and capturing them, storing them in their genome, so that at any point we can go back and recover that information. This technology definitely has applications for experimental biology, so it can be used as a tool for experimental biologists to gather data from cells in the future. But it also potentially has applications in the medical field, where a cell has privileged access to certain components of your body. And if a cell could be gathering information and recording it into its own genome, then that cell could be perhaps even living within your body and getting that information over time. And a clinician could go in and sequence out the genomic locus that has that information and see what's been going on previously. So we're looking at an image of a human hand. We chose a human hand because it was one of the first images that people put onto the natural world. And so since we were encoding images here into the natural world in a different way, we decided to recapitulate one of those images. And what you're seeing is the source image, which is what we started with. We then encoded all of the pixel values into the nucleotides of DNA, distributed them over a number of strands of DNA, and then put them into bacteria, where the bacteria acquired those sequences and captured them into their own genome. After the cells grew for a while, we then sequenced their genomes and were able to reconstruct the image based on the sequencing of the nucleotides that were inserted. And so then what you're looking at is also the reconstructed image from the bacteria after they were sequenced. Here we are looking at an image of a horse, or a movie of a horse running. This was originally taken by Edward Muybridge, and again, it was one of the first moving images that was made, and so we thought that was an appropriate thing to encode. And what we did here was, again, for each frame, break down the pixel values and encode them in the nucleotides of DNA across many different strands, about 100 strands for each frame of the image. And then in this case, we delivered those strands to living bacteria over time. So each frame was delivered on a different day over the course of 5 days. And then we let those cells grow for a little while, and then we sequenced their genomes, and we were able to reconstruct not only each of the frames, but the order of each of the frames, because the molecular recording system that we're using captures the timing information of different molecular events. The next step would be to hook it up to actual biology. All right, so right now we were supplying information in the form of synthesized DNA. In the future, we want that information to come from biological events that are happening within the cell. And so that's the next step.